Dear students, today I am going to explain to you material preparation for transmission electron microscopic studies. Regarding material preparation for transmission electron microscope studies, just I want to tell some more background information about the how to prepare the specimen preparation for block. First of all, to prepare the block, there are some steps are involved. What are the steps? First, one by one I am going to explain to you. The first one is the fixation, then dehydration, then infiltration, then embedding, then polymerization, then sectioning, then staining, and finally observation under the transmission electron microscopic studies. Based on the headings, now I am going to explain to you one by one. First of all, as I already pointed out, the fixation. What is meant by fixation? In the case of the biological science, the fixatives are the two different types of the fixatives. The first one is fixation by means of immersion, and another one is fixation by means of perfusion. So now we are going to adapt it only for fixation by means of immersion. So before going to fix the material, try to understand about what is meant by fixation. Fixation is the preservation of structural and cellular elements in details. Fixation is the frozen slice of life. Fixation should make us a permanent and as a true picture of cell structure as is possible. So these are the objectives, the fixatives. Suppose you are fixing the material. While you fix the material, the fix has a frozen stage. What is meant by the term killing? Try to understand about the meaning of term fixation and the meaning of term killing. As I already pointed out, the term fixation is the preservation of structural and cellular elements, whereas in the case of the term killing means sudden stoppage of the life process with respect to the entire organism and more especially individual cell which will be made up of. So these 2 points you should try to understand. Then fixation. Fixation by means of glutaraldehyde, because there are so many different types of the fixatives are available, but in the case of the electron microscopic studies, exclusively glutaraldehyde. This glutaraldehyde generally for 2% glutaraldehyde. It is available commercially in the aqueous saturated solutions. So how to prepare the primary fixatives? For example, first you have to prepare a buffer solution. This buffer also varied depending upon the nature of the materials. For example, in the case of algae, you have to select crocodile buffer. Some rest of the specimen generally we are used for the phosphate buffer. So first you can take any one of the buffer. So for example, I will tell you crocodile buffer. How to prepare the crocodile buffer? First, initially you take 2.14 gram of the sodium crocodile salt, that is dissolved with the 100 ml of the double distilled water. So this is the fixative 1, that is solution A. Then solution B, take the solution A from, for example, 80 ml, 20 ml of the glutaraldehyde. This is the solution B. So now the primary fixative was over. This is the primary fixative solution. So how to fix the sample? You take the Appendox tube or beam capsule or vials. So take a quantity volume of the primary fixative solution, add 1 or 2 drop of your solutions. In addition to that, fix your sample. The main theme is the sample should be immersed in your solution. Now the primary fixative was over. So the primary fixation by means of the glutaraldehyde, this should be kept in 24 to 36 hours at room temperature. This range is depending upon the nature of the materials. First step was over. Then second step, that is the post fixation. Why we necessary to study the post fixation? Because already we have done the primary fixation. Why again we have to add the post fixation? For the reason is, if the lipid particles, protein particles and some other minute ergostoplasm will also preserved while you are doing the post fixation by means of osmium tetroxide. Then this osmium tetroxide also give the duration and temperature. The duration is 3 to 6 hours and the temperature 4 degrees centigrade, or even you can keep your sample as a room temperature. So the primary fixation and post fixation, that is depending upon the nature of the material, you have to standardize the duration of the time and also the concentration of the time. Then again rinse in buffer. So you are already added the asmic acid, that the excess of the asmic acid should be removed. For that reason we have to rinse in buffer for 3 times each 20 minutes interval at 4 degrees centigrade. After repeatedly washing rinse in buffer solution, then you can go for the next step, which is known as dehydration. What is mean by dehydration? Dehydration is nothing but it is a removal of the water. So the dehydration is a process by which the water particles will be removed from the cells or tissues before or either impregnating, finally mounting the tissues. Dehydration, there are 2 maxims, that is 2 general truth, that is the low intervals long concentration tissue will make a soft, at the same times long intervals high concentration tissue will make a brittle. So these 2 points is very important in the process of the dehydration. In this dehydration process you can take this solvent, for example acetone or alcohol. How to do the dehydration? Dehydration through the upgraded series. First you take the 20% of the acetone. How to prepare the 20% of the acetone? For example, take 2 ml of the acetone is dissolved with the 8 ml of the distilled water. Likewise 20%, 35%, 50%, 70%. Up to 70% give the duration of the time 30 minutes interval at 4 degrees centigrade. So one point I want to tell you in this stage, up to 70% your material should be kept in refrigerator for more than 6 months. Suppose we cannot be able to proceed further steps for subsequently, you can stop up to this stage. Then while you are passed on to 70% to 90%, that material should be always kept in room temperature. Now the sample is 90% acetone. 90% acetone, give the duration of the time 1 hour intervals. Then 100% acetone. 100% acetone repeatedly 3 times you have to give the specimens. For example, 1 hour intervals, 2 hour interval, 3 hour interval. But the material is always kept in room temperature. After that add a dry acetone also. Dry acetone is nothing but in addition to that copper sulfate is added in acetone solution, that is known as the dry acetone. So our main aim is completely remove all the water particles, that is the dehydration purpose. Now let us pass on the another step, that is known as the infiltration. What is mean by infiltration? The process of dissolving wax particles in chloroform by step until reaching the saturation point is known as infiltration. What is mean by saturation point? When the wax which has been already added, that is dissolves, disappear completely. Some more wax will be added in chloroform by step until there is no more wax will be dissolved. That particular stage is known as saturation point. Now how to do the infiltration? In the case of the electron microscopic studies, infiltration, acetone is mixed with the resin. So the resins are usually 2 different types of the resins. For example, high viscosity resin and also the low viscosity resin. Now we can take for the high viscosity resin like Auddeyt. How to prepare the resins? First 75%, 50%, 25%. In this ratio we are considered as 1:3, 1:1 and 3:1. In this ratio we have to prepare the acetone and Auddeyt resins. After prepared these resins, that should be poured into the embedding. So before going to embedding, your resins, that is the prepared resins, should be thoroughly mixed without any air bubbles. If the trace amount of the air bubbles will be there, you cannot be able to get a proper block. So to avoid air bubble, that resin should be at least 6 hours thoroughly mixed with the help of magnetic stirrer. After thoroughly mixed the resins, that should be kept and covered with a black paper and further uses. So this infiltration process was over, then you can go for the embedding. What is meant by embedding? The embedding is nothing but the technique of the tissue is enclosed in the solidified form of paraffin medium. It is known as the embedding. This embedding was usually performed by means of silicon rubber mold or beam capsule or gelatin capsules. So already you are prepared the resin, you take the silicon rubber mold. That silicon rubber mold are also arranged according to the orientation of the samples. You just poured your resins on the performed wells. After added the resin with the help of forceps and needle, you have to arrange your specimens according to the orientation of the samples. So after that, completely you are arranged your specimen without any disturbance. Gently take away the silicon rubber mold and kept in polymerization. So the polymerization by means of hot air oven. This polymerization give the temperature setting 60 degrees centigrade. You have to give the temperature setting, and the time is 24 to 36 hours. This is also depending upon the nature of the materials. You have to standardize the time. After polymerization was over, take away the block and just identified the block whether the polymerization was over or not. After taking the silicon rubber mold, you just how to remove the block. You just make as a V shape. Automatically the flocks are fall down. So after fall down the block, that block as early as the block should be observed on the light microscope. Whether your prepared block, is there any damage or is there any porous or whether fully polymerized or not, everything should be first initially check up with the help of stereo microscope. Once you are satisfied with the stereo microscope, then you can go for the trimming the block. After removing the block, that block should be trimmed with the help of ultramicrotomy. So the block should be trimmed as a pyramid like structures or trapezium like structures. After trimming the block, that should be kept in the block holder. After that, you want to make a glass knives. The glass knives usually for Belgium glass tips are available. These Belgium glass tips, first of all you want to broken down with the help of the knife scorer or knife maker. This knife maker will cut the first your knife should be cut into square angle, then triangle. After making a triangle, that should be attached with the help of the plastic adhesive tabs, and the plastic adhesive tabs plus trough. Along with the trough, that knife should be attached. After attached it, this also should be kept in the ultramicrotomy. There is a provision to fix in the knife holder. After fixing the knife holder, you are already making a block also as a pyramid like structure. So the block should be kept in the block holder, knife should be kept in the knife holder. After fitting tightly, adjust the microtome. Before going to take the sections, the microtome should be thoroughly checked. All the accessory parts, whether it is work or not, you try to adjust manually first. Then this ultramicrotome, there is a main point is without manpower they can able to take this section. So before starting the section, you just adjust it all the parts, then you just press the ultramicrotome system, automatically the sections are come out as a uniform sequence of thin layer. The only thing is you have to select the sections. The sections are come out as a different colors. The colors are ranging from grey color, silver color, gold color, yellow color, orange, purple, green. So these colors are indicating as a 20 nanometer. Each and every color is recognized as 20 nanometer ranges. In electron microscopic studies, generally 60 to 90 nanometer sections are very good sections. So we have to collect silver color, grey color and gold color sections. How to collect these sections? The sections are nanometer, it is very difficult to collect. So you have to take a forceps, fine forceps, that is it is a imported one. The forceps with the help of the eyelaser brush, you gently pick up the sections. After pick up the section, that should be immediately kept in the Wadman number filter paper covered with the Petri plate. So this is the collection of the grids. After collecting the grids, at least 24 hours that should be kept in Wadman number filter paper to completely the moisture should be absorbed. After that, the section should be staining. So the staining here it is entirely different, not for the light microscope studies. In electron microscopy studies, there is a special stain which is known as the positive stain. This positive stains is the combination of uranyl acetate and lead citrate. How to prepare this uranyl acetate and lead citrate? As a saturated solution, for example, you take a known volume of the uranyl acetate salt, that is dissolved with double distilled water or deionized water as a aqueous saturated manner, then further you centrifuged. After centrifuged with the help of the Wartman filter paper, filter it, then that should be covered with the black paper. So now uranyl acetate solution was ready. Likewise, lead citrate solution also prepared as a saturated solution. So these two solutions was ready, then you can go for the staining. How to stain in the grids? So in the grids are very small. So you can take the porcelain tiles. The porcelain tiles also ridges and furrows, likewise they are arranged. So you first with the help of the forceps you take the grids gently placed in the wells, then add 10 to 15 ml of the uranyl acetate, leave for 15 minutes in the room temperature. After that again thoroughly rinsed with the double distilled water. After that grid should be kept in the Wartman number filter paper. After 5 minutes then again you can go for the lead citrate stain. After lead citrate stain was over, then you identified with the help of the transmission electron microscopy. Now I am going to sum up the overall protocol for material preparation for transmission electron microscopy. First you have to collect the material. Collected material should be selection. Selection is the most important point, because whether your specimen selection is which portion, whether root or stem or leaf, and also that particular selected portion should not affect any diseases. That is most important point. After selection was over, then you can go for the primary fixation by means of 2% glutaraldehyde. After primary fixation was over, rinse in buffer 3 times, each 20 minutes interval at 4 degrees centigrade. Then post fixation by means of 1% osmium tetroxide. After post fixation was over, then again rinse in buffer 3 times, each 20 minutes interval. Then dehydration through upgraded series in acetone. After dehydration was over, then infiltration. Infiltration, acetone is mixed with the resins. The infiltration, there is a ratio 1:3, 1:1 and 3:1. In this ratio you have to prepare the resins. This is the infiltration. Then embedding by means of silicon rubber mold or beam capsule. After embedding was over, then that material should be kept in polymerization by hot air oven. After polymerization was over, you just remove the blocks. After removing the block, take semi-thin sections. You are satisfied, then you can go for the ultra-thin section with the help of ultramicrotome. After ultra-thin section was over, the collected section should be stained with the positive stains. After staining was over, then you observe under the transmission electron microscope. So this is the protocol for material preparation for transmission electron microscopy. Finally, I would like to tell conclusion. What is the essential for the material preparation for electron microscopy studies? For example, to find out life cycle patterns like haplontic life cycle, diplontic life cycle, haplodiplontic life cycle, alternation of generation life cycle, all these things will be assessed by means of qualitative ultrastructural studies. Another point is, for example, in anatomy, nodal anatomy, floral vasculature, embryogenesis, corposporogenesis, tetrasporogenesis, spermatogenesis, all the developmental studies also you can easily trace out with the help of uniform layer of thin sections. And another point is, ultrastructural studies will assess the systematic position of particular family character also.